Today's webinar from Dr. Owen Fahy will focus on the National Metabolomics Repository, an international resource for sharing metabolomics data, metadata, tools, and more. Before introducing our speaker, I will quickly review a few logistics. Should you experience technical difficulties, you can contact us through the questions box in the expanded control panel, or by phone or email. If you need to view live closed captioning, please click on the link that will appear in the chat box. At the end of Dr. Fahy's presentation, we will allow ample time for questions. If at any time during the presentation you have a question, please type it into the question box, and I will ask it on your behalf during the Q&A session. So at any time you have something pop up, feel free to type it in there, and we'll get it in the queue. So it is my pleasure to introduce Dr. Owen Fahy. Dr. Fahy has a Bachelor of Science in Biochemistry from the University College of Galway and a PhD in Chemistry from the University of British Columbia, specializing in the structural elucidation of marine natural products. He completed a postdoctoral appointment at the Scripps Institution of Oceanography in the University of California, San Diego. He has over 15 years experience in the biotechnology industry in the areas of organic chemistry, drug target discovery, molecular biology, proteomics, genomics, and informatics. He has been involved with the Lipid Maps Consortium since 2003, where he serves as a project coordinator for the bioinformatics efforts. His work in this area includes development of a lipid classification system as a member of the International Lipid Classification and Nomenclature Committee, design and development of database infrastructures for lipidomics, development of mass spectrometry software for lipid research, design of novel lipid structure drawing tools, and development of integrated pathway tools and resources. Since 2012, he has served as the Bioinformatic Project Coordinator for the National Institutes of Health Metabolomics Common Fund Program and the National Metabolomics Data Repository at UCSD. He oversees the Metabolomics Workbench website, which acts as a national and international repository for metabolomics data and metadata, and provides analysis tools and access to metabolite standards, protocols, tutorials, training, and more. So we welcome Dr. Fahy to today's webinar, and I am going to turn it over to him.
I want to give you an overview on the work we've done with the National Metabolomics Data Repository, NMDR, I'll call it from now on, which has been in existence for about since 2012, so almost 8 years. It's housed at the University of California, San Diego, in conjunction with the San Diego Supercomputer Center. And this is our main portal, the Metabolomics Workbench. This is really the grand central station for all of the data repository data and other applications. So as you can see right now, we have well over 1,000 publicly available studies, and a number of these will be, more of these will be available when their embargo dates come around. So on the right-hand side, you can see the main groups associated with the whole project. There's a coordinating center in the University of Florida, the M3C as we call it. Our group at UC San Diego, the Metabolomics Workbench, where the actual repository is housed. And then two other major core groups, the compound identification cores, the number of universities involved in that effort, and also the data and tools cores, which do a lot of software development in the area of metabolomics. So this is an overview of the growth of the NMDR. We actually started getting submissions back in January 13th, so that's 7 years ago. And this graph shows the profile of the increase in studies. We went to an online deposition system in 2015, and there was a spike up there. We went to a more automated system in 2017. So the current rate of deposition is probably about 20 new studies per month. So we're holding steady with that. This is an overview of the major taxonomic categories. No surprise that human and mouse are by far the largest piece of the pie in terms of species. But we have many other mammals as well, rat, monkey. We have a pretty large section for bacterial studies as well as a number of minor organisms. So it's pretty well represented. Many of the studies are related to some kind of a disease outcome, and cancer again is probably the largest disease category, followed by diabetes, obesity, and many others. So in the other category, you have many rare diseases in both animals and plants in some cases as well. In terms of sample source, we track that. Blood is the largest contributor, also cultured cells, and many other human and mammalian organisms dominate this sector. This slide shows an overview of the inputs and outputs into NMDR. So we gather online data submissions, so the experimentalists will fill out various online forms and upload their data. We also get the raw data. Pretty large, it's stored in an FTP repository. And then on the bottom side, we have all the outputs. Obviously, the website is a major browsing, searching tool. We have statistical analysis tools, which I'll go into. We have workflows both for targeted data and untargeted data for mass spec and NMR. One can download the targeted and untargeted measurements. One can download the raw data for reanalysis. And there are also other options such as the MWTab file, which I'll talk about more in detail, and also a REST service, which is very powerful in terms of automating the NMDR data into other workflows. This is an overview of what happens to the experimental data in terms of where it goes and what we do with it. So on the left-hand side, we have studies of name metabolites, usually targeted data, where all of the metadata and the results go into relational databases. Then on the top right, we have the situation for untargeted data sets. So these would typically be large LCMS data sets where we use a large file format instead of a relational database. And we curate that data and store it in a file-based system. But the bottom line is that we have the same analysis tools available for both relational database workflow and the big data file-based workflow. So in terms of user experience there, it's very, very similar. So this is the online submission overview. People submit a study. They have to fill out a number of forms to enter all the metadata. So what is actually going on is a special file format called an MWTab file is being created stepwise as the submitter enters the data. And that then is curated by the NMDR group. And once it's ready for release, and once the any embargo dates are passed, it becomes available on the workbench. That MWTab file can be read in many different formats by our web infrastructure to do all kinds of things in terms of viewing, searching, and statistical analysis. So this is the MWTab format. So what we view it as a common currency for metadata and data sharing. If you're familiar with the UniProt file format for proteins, it's somewhat similar in that it has tags on the left-hand side which describe the text. So it's human readable, but it's also very computer friendly in terms of being able to be parsed out. We have all these metadata sections and also the named data the results as well. And so it's basically a single file format that will encapsulate all of the data, with the exception of raw data. So an online data submission, this is our page for submitting data. The user has to register with the workbench, and once that's done, he or she can start submitting a study. So the workflow for submitting a study goes through different sections where the information about the study, the overview, what the subjects are, the study design, which is crucial. So by study design, I mean, what are the experimental parameters? Is it disease versus what samples are disease, what samples are control? Is it a treatment study? Is it a time course study? So that's obviously very important to delineate that in terms of analysis downstream. So various decisions have to be made through the workflow. Is it a mass spec study? Is it an NMDR study? How many chromatography methods are used? How many mass spec methods? And so on and so forth. And once that's completed, then the data can be curated. So this is just one example of a submission form. And so all the items in pink over here are mandatory, and many of these are filled out once you register, like name, address, institution, all of that stuff. So there are a number of controlled vocabularies in terms of pull-down menus, or one can choose a new one. So once all that is complete, and once the study is curated and put on the workbench, then it's available to the general community. And if you're following along online, you can see this main page for browsing and searching the studies. So one of the first things you might want to do is look at all of the studies, the summary of the studies. So this is a big long list right now. It's about 1,100 rows long. So you can, on the right-hand side, you can actually download the raw data. On the left-hand side, you can click on it to view a study of interest. So, this is one study of interest, the first one, study one. It has a number of links here where you can look at samples, the named metabolites, download the named metabolites, download this NWTAB file format that I talked about earlier, either in text or JSON. And importantly, you can perform statistical analysis on that particular study of interest. So one thing we've tackled right up front is the issue of metabolite nomenclature. So why do we need this standardized reference nomenclature? Well, there's a huge amount of diversity in reporting names of metabolite species. So pretty much every lab has their own way of naming metabolites. And this is a huge problem in terms of data sharing and comparing and contrasting studies. So we had to tackle this up front. So what we've done with all of these studies on the workbench is basically to distill all of the 220,000 named metabolites that we've seen so far to come up with a chemistry-centric database. So by that I mean we want to have a list of metabolite names in a standardized format that one typically detects by mass spec or NMR, as opposed to pulling them in from some database. So we put a lot of work into this. So every study that comes in the door, we map the user submitted names to this RefMet standard reference. So there are basically 4 types of annotation groups. There are cases from targeted studies where the structure is actually known, doing the stereochemistry and double bond geometry, like PGE2, folic acid, so on and so forth. And there's a second group of metabolites where you have known regiochemistry, but not stereochemistry and bond geometry. So an example would be 12P. So you don't know the chirality at the C12 center, or say 3-hydroxy tetradecaenoic acid. And the third group are partial structures. So for example, if you do mass spec on a phospholipid and MSMS, and you can determine the masses of the change, say 16:0, 18:1. You don't know what the SN regiochemistry is. You don't know where the double bonds are, but you have some information, like in this example here. Then the fourth group is where you have just some composition data. So this would be from just MS1 high resolution studies. So for example, PC34:1 or ceramide P42:2, where you know the bulk, some composition in terms of carbons and double bonds and the metabolite class. So basically, we have to deal with these four different groups because that's what comes in the door in terms of study analysis. So this is an example of what we're dealing with in terms of the heterogeneity of names. And it's actually a lot worse than this because people put suffixes after the ends of the metabolites, charge state, ion type, M plus H, M plus Na, et cetera, et cetera. So what we're doing is basically distilling all of these different diverse names into one common format. So we have a database, RefMet, and it's available online and downloadable, and a companion metabolite database on here on the right-hand side. So for group 1 and group 2, these are actual structures that you can draw. So those structures are actually stored in a metabolite database. For group 3 and 4, those are just annotations. Those cannot be defined as a structure. So those don't have any analogous entry in the metabolite database. So this is the page for RefMet. You can browse, search, download RefMet, and convert metabolite names to RefMet nomenclature. This is a view of the search tool. You can search by Vinci key, name, mass, formula. You can do a substructure search if you want, and you can download the whole thing too. So this is an example of a detail page for RefMet. So this is an exact structure, cholic acid. Quite a number of options here in terms of searching by formula, searching by other enantiomers, diastereomers, using the first 14 characters of the Ichi key. So that's quite useful. It can be viewed on any type of browser, iPad, iPhone, or whatever. We recently merged metabolite data with the KEGG reaction data for human pathways. So for a metabolite of interest that is a member of a human pathway in KEGG, you can drill in deeper in terms of that particular reaction or that particular pathway through the KEGG infrastructure. We also link all the RefMet metabolites were applicable to a database of mass spectra called MONA. This is the Mass Bank of North America. So then this is a very useful resource for accessing public domain mass spectra. So I think there's about 100,000 entries that are relevant to RefMet in MONA. So that's a useful resource. So this is typically MSMS spectrum. We also have a tool for doing your own harmonization. Like I said, for every study that comes in, we do this mapping from metabolite user-supplied metabolites to RefMet, but you can also do it online yourself. So this is an example. You just copy and paste your list of metabolites. Let's say you have 200 and run it, and it'll match as well as it can to the RefMet database. Obviously, if you have some very obscure metabolite name, it's not going to find a perfect match. So this is an example of what we're up with. Let's say one searches for LPC 16:0, lysophosphatidyl-choline 16:0. It's very easy to do that, having this standardized nomenclature. But on the right-hand side, you see what people actually submitted. So if we did not have this here, it'll be quite a laborious procedure to pick out all the studies that have LPC 16:0 because there are so many different synonyms. We also map all of the metabolites to metabolite classification system. For the lipids, we use the lipid maps classification, which has been available for many, many years. And for the non-lipids, we use classifier classification developed by the Wishart group in the University of Alberta, which can handle any organic molecule. So we curate that and merge the two together. The bottom line is we have a hierarchy of classification system for all of the RefMet metabolites. So you can do something like that. Look at a pie chart by superclass, our main class, or subclass. So basically, the RefMet commands a central role on the Metabolomics workbench. Very importantly, we can compare and contrast metabolites detection in many different studies because we're speaking the same language, so to speak. We can classify the metabolites and, for example, migrate those to the Lipid Maps portal. So we have a collaboration with Lipid Maps where you go to the Lipid Maps website and have a portal into the workbench for all studies containing lipids. I think there are several hundred workbench studies which contain lipids, and you can migrate through the classification system there to find the lipids of interest. We can also link to metabolite structures, as I mentioned previously. That's a very powerful thing. We can do substructure searches and link to other databases. We can link to other ontologies. For example, the Lion Web ontology at the University of Utrecht, I'll talk a little bit about that later, has a way to map other metabolite ontologies like number of carbons, double bonds, membrane permeability, subcellular localization, et cetera. So using the RefMet system, we can link into that. We can also link to metabolite pathways. We have links to SMPD, that's HMDD, that's the Human Metabolome Database, and associated pathways. Also the KEG system in Japan, and also the Reactome system in the UK. By linking through the metabolite names to the corresponding substrates and products in these pathways. So the bottom line is we can map an incoming study, take the metabolites and map them to pathways. And I'll scoot here if I can. Okay. We also have what we call our collaboration subsets. So for example, we're working with the MotorPack group, which studies the importance of metabolism in exercise in both humans and rodents. So the MotorPack group is doing mass spec analysis and are taking a subset of RefMet for their uses in terms of their human and rat studies to exercise. Trying to advance the slide. Oh, there we go. Okay. So the positive outcomes, we have a unifying nomenclature, ability to perform comparative analysis across studies, linking to other structured databases, comprehensive classification system, integration with biochemical pathway tools. So getting back to the analysis of data. So for any particular study, I showed you a link a few slides back to a statistics toolbox. So one can do many different types of analysis on the study of interest, and these are organized into groups in terms of pie charts for metabolite classes, normalization and averaging, things like volcano plot analysis, ANOVA analysis, clustering, PCA, LDA, and random forest, and mapping to biochemical pathways. So there are many options depending on whether it's a mass spec study or an NMR study to do your own analysis for a study of interest. So this is an example of one study of interest. This is a study on the effect of glucocorticoids in dogs, long-term and short-term. So if you go to this particular study, 1140, and do volcano plot analysis, for example, first thing you want to do is, set it up in terms of what are going to be your two different groups. What are you going to compare? So, for example, before and after treatment with this particular drug. And many of these studies have several different analyses. So this one has one analysis for phospholipids, DAGs, and cholesterol esters. It has another one for ceramides, another one for fatty acids, and so on. And then you run the volcano plot and get all of these different views of the data. So, for example, this is a class enrichment analysis because, like I say, all the RefMet names are mapped to classification. You can do bubble plot analysis and point and click on the different subgroups and so on and so forth. Similar tools for the other, similar views for the other tools. One other way of looking at the data is through the bubble chart axis. So this is one of the first level items. People want to know, okay, who has done studies on rats, for example, I want to know what metabolites we can detect in liver or urine or kidney and so on and so forth. Might be interested in one particular disease. People might want to see all the studies related to, say, tuberculosis or obesity or whatever. Or you may be interested in a particular metabolite class. So this is just an easy way to access the studies for a particular area of interest. So once you click on any of these bubbles, you get a list of studies pertinent to that selection. We have the database, metabolite database of 80,000 molecular structures that I alluded to earlier on, fully searchable in multiple different ways, by substructure, by INCHI keys, PubChem, IDs, and cross-reference to many other publicly available databases. So this is just an example of one particular metabolite. We have all of these external database links and links if that metabolite is found in any of these human pathways, links to those particular pathways of interest. So this is an example of exploration of pathways of interest. I think this is a reactome example for alanine. We have a companion gene protein database, human gene protein database. This data was pulled from NCBI Entree and also Swiss Protein Uniprot for genes that had some relevance to metabolism, either through a Go annotation or some other summary annotation. So one can browse this through the databases menu on the home page and get access to many different features of the genes and proteins, the actual RNAs, the protein sequences, and so on and so forth, and link to the relevant metabolites for a particular pathway of interest. We also have a roll your own tool search. So if you want to, actually this is a mass spec search. So if you want to do mass spec analysis on a database, you have a number of different options. You can search the RefMet database. That's about 95,000 annotations. You can search a virtual database of lipid classes, and you can also search the metabolite database that I just talked about using a number of different ways. There are also other options like searching all our untargeted MS data by MOZ retention time, also running the RefMet search as a Shiny app. So this is an example of the user interface for searching RefMet. You can enter your list of precursor ions, limit the search by class, select whatever ion adducts you're of interest. So you'll be either looking at positive ions or negative ions, one or the other, and then running the search. You can also search the untargeted data. So we're sitting on a very large amount of untargeted LCMS data. We're looking at over probably 2 million M/Z features to date from well over 400 studies and 800 MS analysis. So most of these data have not been assigned to a particular metabolite. So typically we might get 10 or 20,000 features in terms of M/Z and retention time values from a single study. So it's a lot of data. So for example, if somebody had an ion, an interesting ion that they wanted to see if somebody else had also found it, or whether there are any other association with it, you could search this very large database. So this is just a search on that particular ion, 657.48. So you can see down in the bottom here, we've merged cases where people have actually identified that ion from data. So that might be a clue as to what that unknown ion would be, and obviously you have to match what the chromatography conditions are. Was it the same system, same solvents, et cetera, same ion type. So I think it's going to be a very powerful resource because of the size of the database, and as we continue getting more named annotations, it's going to give more information to the end users. So this is an example of drilling down into the metadata for a particular analysis where you can get all the details about the chromatography for that particular ion. Probably one of the most useful tools on the workbench in terms of finding stuff is what we call the quick search tool in the top right-hand corner. So if you're on the home page of the workbench, you'll see it up in the top right corner. So what this does, you enter a metabolite name, it'll try to do an exact match on RefMet, it'll do a partial search on RefMet. If it's a molecular format, it'll detect that and do a molecular format search. If it's an INCHI key, it'll search that. You can search it with a neutral mass, just give it a number. It also does a number of other searches on the metabolite structure database. It'll search that metabolite name in all the NMDR studies. So if it's found in any study, it'll show you a list of those studies of interest. Also search the metabolite classification hierarchy and the resources on the website. So this is an example of a search on the word alanine. So it gets one exact match on alanine. It'll get many other matches on the metabolite database. It gets 400 hits on the studies because alanine is found in so many studies, and so on and so forth. Here's an example of glycerol. This is why exact match is so important. If you were to try to search a metabolite database with a substring of glycerol, that word is found in so many metabolites, so it's very difficult to fish that one out. We have a tool where you can load and analyze your own data set. Let's say you have a data set in a spreadsheet or on a text table. You can do all of this statistical analysis without having to submit it to the workbench as a proper study. So you can kind of load your own data file, do your own analysis, and there are tools for that on the main page too. So it involves a portable toolbox design using R statistics on the back end, PHP and JavaScript. So it's we've migrated it to a number of other online resources too. We also have a cloud computing collaboration with the San Diego Supercomputer Center, where we're putting these statistics tools in Docker containers, running them on Jupyter notebooks, and they've just become available using my binder free service for Jupyter notebooks. So you might want to check out the Analyze menu item on the home page for this information. This is just a quick overview of the Jupyter tools. It's an example of Python module, volcano. This is the Jupyter Notebooks page that I was talking about. So it's online now. So you have options to download these tools from GitHub or run them on Binder. We're collaborating with the MC3 Center in Florida. Migrate a lot of our data, metadata, to their website, and they have a portal to be able to find study information as well. They don't have the actual results data, but you can get a lot of information there too. We have a collaboration with the CHEER group, which studies children's health and exposure analysis. So we have a specialized portal to work with the CHEER folks in terms of the study that they submit. We also have a MotorPack collaboration that I talked about before to study the effect of physical activity on metabolomics. That's going quite well. They're using the RefMet system now. This is a view into the MotorPack data dictionary, which is basically a subset of RefMet. It's about 2,000 different metabolites. We have a collaboration with Lipid Maps in Cardiff University, where we have a portal into all the metabolomics workbench studies that contain lipids, and many lipids are involved. Actually, 65% of all identified metabolite species are lipids, so it's a huge piece of the pie. And this is the portal from the Lipid Maps website to the studies on the workbench, numbers of lipids, what the lipids are, and then you drill down and go to the workbench for that study of interest. We have tools for summary statistics where we can answer questions like summarize all of the data on the workbench by disease, by species, by sample source. So somebody's interested in what metabolites are available from mouse in blood, for example. What metabolites have been detected in studies related to diabetes, and so on and so forth. So basically, we have a front end here where one can decide, set up menus for disease, sample source, species, analysis type, and so on and so forth. These pull-down menus give the numbers of studies on the workbench related to that particular item, whether it's disease, sample source, so on and so forth. So it's basically a query builder, and this is the output. You can, this is, for example, for the tuberculosis study. So you can drill down and get all the data for that particular metabolite, for those particular studies, what pathways are related to, what the actual structures are, if they're a real structure, what the studies are, and so on and so forth. And for example, compare and contrast. This is the example for tyrosine. All the studies for tyrosine, and the two studies up on top here list all the studies that have tyrosine related to diabetes. So you can compare and contrast studies using these tools. This is an example for those for two particular studies, study 348 and 349. So you have all the metabolites found in one but not the other, and vice versa, and metabolites found in both. We have a REST service to draw information from the workbench. It covers many different aspects: compounds, studies, samples, metadata. It's a very fast way to pull data from the workbench. We have an interaction, like I mentioned, with the Lion Web, where we can use the REST service to pump data to this website, where in turn that data is analyzed in an ontological manner in terms of what the different features of the metabolites are. For example, numbers of carbons, double bonds, whether membrane fluidity, so on and so forth. So this is just a good example of how we can merge and migrate data from one website to another. This is another example of a REST service where we're working with Scripps Research Institute in La Jolla, in San Diego, where they're interested in mapping MS data to pathway analysis by this Lummi-Chog meta-XAMS analysis. So again, they use the REST service to pull data from the workbench and use that as an input for their pathway analysis tools. We share data with our European colleagues. Metabolome Exchange is a European-funded project where they're taking metabolite studies from all over the world, workbench from EBI in Europe and other resources, and putting them available and making it a searchable website for metadata, and there are obviously links to workbench from there. And another example is the EBI Omics Discovery Index. Again, it's a integrated platform for merging data sets from around the world, and as soon as a workbench study is made available, it also becomes available on this Omics Discovery Index. I'm almost finished now. Metabolite Workbench is fair. It's findable in terms of assigning a DOI to every single study, and that's the item that's usually published, and it's a persistent identifier for that particular study. Accessibility, we have the workbench, we have the REST service. Interoperability, which is really important. We have the MW tab, common currency format for metadata and data. Obviously, the RefMet standardization lecture is the key deliverable too. Reusability is very important. We allow download and reprocessing of all of our data. We're going to migrate all of these data to the cloud platform, and we've already started that with Jupyter Notebooks and MyBinder applications. So we're going to ramp that up quite a bit. So in terms of the ongoing efforts and the summary, we have currently 1,260 studies. We anticipate this will grow considerably as our efforts with other large data generating organizations such as MotorPack and Cheer ramp up. We have the RefMet database that we're continually curating and adding to. We have the REST service, a very important interface into NMDR data. Our collaborations are ongoing and growing all the time. We're making outreach efforts to other metabolite class-specific portals, such as lipid maps and other groups that are interested in one particular group of metabolites, and I think we can do a lot with them. And that is about it. I just want to give an acknowledgement to my colleagues here. This group is led by Shankar Subramaniam. He's a professor at the bioengineering department at UC San Diego. We have Kanan and Manish Sud, who are the main curators of data. Sonal is our web master person. Srini is working with human data now because we want to ramp up our efforts in terms of curating human data with our efforts with our other colleagues. And Kevin and Christine at San Diego Supercomputer Center are leading the effort in terms of making our resources available on the cloud. So thank you for listening, and I'd be glad to entertain any questions and comments.
Thank you so much. We did receive questions, and I think we have time to answer them. So I'm just going to start reading them, and you can feel free to answer them. The first one is, what is the largest population submitted to your data repository?
In terms of numbers of samples?
It says the largest population, so I'm assuming that that's all the context that I have.
Off the top of my head, I wouldn't know, but we have several studies where there are maybe 200 or 300 samples from human subjects. So I think those will be the largest. We're not talking about thousands, definitely not.
Great.
I would say, the typical range will be anywhere from 10 to 300.
Thank you. The next question is, can you use the workbench to standardize metabolite names without submitting your data to the repository? In other words, can you use the tools on the workbench without submitting data?
Yes, yes, you can. So if you wanted to standardize your metabolite names without submitting to the repository, you absolutely can do that. Go to the RefMet link, and there'll be a link to map metabolite names to the RefMet nomenclature. And you can just save that as a text file. Yeah.
Great. Next question. Does the software perform peak finding and picking from the raw data?
No, we don't. We don't deal with raw data. We deal with process data. So peak picking and peak finding, you'd have to use XCMS or some other tool like that. So we don't actually do that. Although we're working with other groups in the data cores who are doing that. But as it stands right now, no. We take in raw data, but we don't manipulate it.
Thank you. Next question. Are RefMet ID and HMDB ID the same?
No. So the RefMet name is actually the ID. HMDB is something totally different. So RefMet is an analytical chemistry-centric database. Like I said, it deals with more than just exact structures. It deals with partial structures and bulk compositions as well. We have 4 different groups. So no, RefMet is a totally separate entity, but the metabolites, the actual structures are linked to HMDB. So we have a mapping, ability to map where applicable to HMDB and other databases.
Next question in that same regard, it says, other than the species, how is this advantageous over HMDB? Is there a list of metabolites by species and organs that can be downloaded in Excel?
So if you're interested in one particular species or one particular organ, you can go to the bubble plot analysis. That's one easy way. You can go to that Metadata analysis that I showed near the end of the presentation, where there are pull-down menus for both species, sample source, and disease association. And then you will get a list of studies pertinent to that set of criteria, selection criteria. Then you can download all of that data for those studies one by one. So yes, you can download all the names, metabolites for all the studies of interest once you've made your selection.
Next question, can you use the tools on the website to examine pathways without submitting data?
You can use the tools to see. Do they mean their own data?
I assume so, yeah.
I imagine these, let's see. I don't think we have a way to map them onto pathways right yet. There is a toolbox where one can upload their own data, do statistical analysis. Off the top of my head, I don't remember whether we've implemented the pathway mapping to that. You can definitely look at any study of interest and map that data to pathways, but I'd have to check on that one. I don't think that's been implemented yet.
Okay, I think we have time for one last question, and we received one. Are healthy subjects included in the studies submitted, and can they be gathered in one pool?
Almost all well-designed studies would have, healthy subjects as controls. So I don't know what they mean exactly by one defined pool. Would that be across studies? Definitely within a study. Typically,
I think they want to. My guess is that they want to pull all the healthy subjects across to the data that you have together to use. That's my guess, unless they clarify.
there isn't a one magic button that you can push to pull in all the healthy subjects. No, but you could definitely do it one by one by identifying the studies of interest, depending on what disease you're interested in, and downloading the data for the healthy subjects in there.
Great, and they did confirm that was the question they were asking, so
Yeah.
we answered the right question. But time is running short, so I do want to wrap things up, and I want to thank you all for your attention and your active participation in today's NIH Metabolomics Interest Group. And we would also like to take this opportunity to thank our presenter, Dr. Owen Fahy, for an excellent presentation. Thank you so much, Owen, for spending some time with us today and explaining a little bit more about Metabolomics Workbench. I think we all have appreciated it greatly. And then we welcome all of you to provide feedback to inform our future webinars. So thank you all, and everybody have a great day.